Okay, so now we are live on air. So this is on our YouTube channel and also the Google Hangout window. So we have folks online. Actually, Laurel, what state are you in now? I think, oh, Laurel should have worked. I think she's in Michigan now, or Iowa. I can't remember. I know you moved north. We've got Minnesota, we've got LSU on the line, and we have our speaker seminar, Esther Angert, up in New York State, enjoying some unseasonably cool weather this last few days.
It's sunny right now, so we're
sunny,
good.
Yes, but at least cool. All right, and so Esther will be talking to us today about big bacteria and some of the things that her lab has done with them. And as previously, we will ask everyone to mute their microphones, and you're welcome to shut off your webcam if you don't want us to see you picking your nose. And we'll pause halfway through for questions within the Hangout, and you can also ask questions through Twitter to @microseminar. The hashtag can also be used, and I will try to keep on top of that in order to ask questions if you're not here with us. So we can have Esther do your share screen, and you can get started.
All right. And all right. Thanks, Jen. Oh, can you see me? I think my camera went out.
So we won't see you. We'll just see your talk.
Okay. All right. That's why the camera went out. Very good. Thanks, Jen, for inviting me to present in this seminar series. You know, looking at the list of speakers, they're all fabulous microbiologists, some of which I know, some of who I just know by reputation, and it's a real pleasure for me to be among this illustrious group. I noticed in the seminar titles that there were some people talking about extremophiles, and I'm going to continue on that thread and talk about a different kind of extremophile. I'm going to talk about big bacteria, focusing primarily on the big bacterium that my laboratory studies, but I'm going to try to bring in some information from other organisms as well. So my laboratory studies two groups of intestinal bacteria. Our model organisms are Epilepicium species type B, which is a large bacterium that's found in the intestinal tract of the surgeonfish Nasotonganus. We also study Metabacterium Polyspora, which is a large bacterium that's found in the gastrointestinal tract of guinea pigs. We like to study these two groups because learning a little bit about the symbiotic association in one system oftentimes informs the other system. And we're also interested in the cell biology of these organisms. If you look at these two micrographs, you can see that there are some really gross morphological features that the two groups share, and that's the production of multiple intracellular bodies. In the Metabacterium image, you can see these four-phase bright objects, and in Epilepicium, you can see these two large structures internally. Those are offspring, and we've been interested in where this unusual form of reproduction evolved from. So I'm going to start my talk out to...
Sorry to interrupt you. Can you do full screen on your slides? Because right now we can see your PowerPoint window.
Say that again. Do a full screen. Is that not full screen?
So right now we can see the entire PowerPoint window.
Oh.
I know you have two monitors, so it might be...
Maybe I'm doing it on the wrong...
Yeah.
This is one of those things, right? Because it's in full screen for me, and that's why I'm kind of confused. Let me try a different approach here. Format. View slideshow. Did that work?
I think something's happening, but we can still see the full PowerPoint background.
Ah. Do I have to do any kind of system setup change? Because I'm seeing the full slideshow on my end.
Nope. Looks better, I think.
Do I have to do anything like... Do I have to do a display thing, do you think? No?
You may. You have to switch your monitors.
Okay. It's not giving me... Show mirroring options. Okay. Did that work?
No, I think so. I think we're seeing your other monitor still.
Yeah, I'm not attached to any other monitors.
Oh shoot.
Yeah. Ah. Any ideas from the audience?
The other thing is, if you, like in the PowerPoint window, if you can get back to that original PowerPoint window and then just sort of change the view so it's just the one slide.
Gotcha.
Like so we can see your your slides outline. Yeah, okay. And then just like enlarge that and then. Yeah.
Oops, no, don't want to do that. Ooh, that's too big. Is that big? How's that? No? Yes.
That's good. And if you maybe just shrink the bottom notes window and we can make sure we see the whole slide. Sorry. Okay.
Does this, there's a broadcast feature, is that going to help? No. Never mind. Okay.
Okay, let's go with it.
Okay, very good. So let me, now how do I advance? Okay. Ah! Now I'm having trouble advancing. Sorry about this. There we go. Okay. All right.
Now for the line 27A.
Are we ready? Ah. Sorry about all these technical problems.
No problem, and if you want, we will delete it off the recording.
Okay. Very good. Okay, let's try this again. So my laboratory. Oh. Yeah, I'm really having trouble here advancing these slides even. Is that big enough? I think most of my slides are pretty bold.
Yeah. Okay, that's great.
Okay, I'm going to move forward then. So we studied two groups of intestinal bacteria, and today I'm going to focus primarily on talking about members of the genus Epilepicium, which are surgeonfish intestinal symbionts, and primarily talk about Epilepicium type B, which is our model organism for studying these large bacteria. I'll tell you a little bit about Metabacterium Polyspora as well. So I'd like to start by giving you an introduction to these organisms in case some of you don't know Epilepicium or Metabacterium, and tell you a little bit about their discovery, their initial characterization. So Epilepicium was first described by a group of fish biologists who were studying surgeonfish in the Red Sea, including the brown surgeonfish Acantharus nigrifuscus. When they opened up the intestinal tract of these bacteria and looked inside, they saw some pretty amazing large microorganisms. This is a short movie that I'd like to show you that shows you the what you might see if you took a little bit of gut fluid from a surgeonfish and put it on a microscope. This was just shot with a regular old commercial, consumer grade Canon camera held up to an eyepiece of a Zeiss microscope using a 10x objective. And what you might see is rolling around in the fluid, swimming through the fluid channels that were formed between the cover slip and the slide are these large microorganisms swimming along pretty happily, but they're enormous, and in parts of the gut, they're pretty much the dominant biomass of the intestinal system. Individuals of this group of microbes can reach tremendous lengths, such as this individual, which was 600 microns in length. They also exhibited a number of unusual features, including the production of multiple internal structures. In this particular individual, I hope you can see that there's one structure here and a second structure there. Those are offspring cells or daughter cells. Offspring are initiated in the tips of a mother cell. They grow inside the cytoplasm of the mother cell, and eventually they burst out. These are two Nomarski differential interference contrast micrographs showing offspring emerging from a mother cell. And I hope you can see this tear in the mother cell envelope, and the offspring cell is arching its way out of that tear, and then this cell is just a slightly later stage of the process. This group of large microorganisms exhibited a number of features that appeared to be unique and didn't fit in some of the classically described morphologies of protozoa. So Lynn Montgomery and Peggy Pollock described this organism in a publication that came out in the Journal of Protozoology in 1988, and they gave this group of organisms the name Epilepicium fischelsoni. Epilepycium means guest at a banquet of a fish. About the time that these publications started emerging about Epilepycium, Kendall Clements, who was a graduate student at James Cook University in Australia, studying reef fish and their nutritional ecology, he was curious whether or not he could find similar microbes in the intestinal tract of some of the tropical fish that he was looking at. So he did an extensive survey looking at and classifying microorganisms found in the intestinal tract of a variety of different reef fish based on their morphology. So he found a number of different ciliates and flagellates, but then he found this group of large microbes similar to Epilepycium that he grouped into 10 morphotypes based on their overall cell shape, their size, and their mode of cellular reproduction. And he published that survey in 1989 in Marine Biology. This next slide just summarizes the results of what Kendall saw. He looked most extensively in other surgeonfish. The surgeonfish family has about a little over 80 known species that have been described, and that family is divided into multiple genera. He looked at members of all of the genus, the genera of surgeonfish, and found that there were a number of these large microbes in the intestinal tract of members of the Acanthrus species, such as Acanthrus nigrifiscus that I showed earlier. He also found them in Tinaquita species, Nasospecies, Zebrasoma species. But he didn't find them in all members of a particular genus of surgeonfish. For example, this guy down in the lower left-hand corner is also an Acanthrus species, but it doesn't harbor these large intestinal microbes. Kendall found that these microbes, which are shown here, this is sort of the variety of microbes that you can find in surgeonfish guts. These are all micrographs shown at the same magnification. The amorphotype that Kendall described would be represented by that earlier slide of the large epulo that practically filled the screen. If it was on this particular slide, it would pretty much fill the screen as well. The second largest morphotype is the B morphotype that we study. These guys get to be about 300 microns in length and don't exhibit any evidence of binary fission, and instead rely on multiple internal offspring formation for reproduction. There are also a number of smaller morphotypes that don't appear to use binary fission, but some filamentous types as well that do show evidence of binary fission here, and I hope you can see it here as well. So he found these large microbes associated with surgeonfish, primarily surgeonfish that were eating a lot of algae, eating a lot of detritus, or surgeonfish that were omnivores. So he expanded his survey to look at other herbivorous reef fish that were feeding side by side with surgeonfish on the reef, members of the rabbitfish family, Siganids, and the parrotfish family, the Scarids. He never found these large microbes in their intestinal tract. So it appeared to be a very specific symbiosis between these large microbes and their surgeonfish hosts. However, with the unusual complement of characteristics seen in these microorganisms, their large cell size, their unusual mode of reproduction, and maybe a less obvious feature, their motility, very similar to ciliates. However, these cells don't have cilia on their surface. Instead, they are covered with fine filaments that look more like bacterial flagella. So with this unusual complement of characteristics, it was unclear where Epilepicium or these other large microbes fit in a larger phylogenetic context. This is probably a familiar tree for most of the people watching today. This is a phylogenetic tree based on small subunit ribosomal RNA sequence comparisons. From these types of analyses, we know that extant life forms, cellular life forms, fall in three major domains: the bacteria, the archaea, and the eukarya. So it was unclear if Epilepicium was in fact a eukaryotic microbe, if it was a bacterium, or if it was something totally unique to biology. This is about where I came in on the scene. So I was a graduate student at Indiana University, and when I asked to join Norm Pace's lab, he just happened to have some surgeonfish gut contents in his refrigerator that had been sent to him by Kendall Clements. So he pulled some of those samples out, we looked at them under the microscope, and he said, hey, how about if you work on these organisms? And little did I know at the time that it would become kind of a lifelong obsession for me. So what my task was to take gut contents, surgeonfish gut contents that were fixed in ethanol and shipped off to Indiana, and my task was to get some phylogenetically relevant information from these enormous gut microbes without first having to devise a means of growing them in the laboratory. At the time, it was kind of challenging, but these methods have become routine, and lots of people are doing this now. So I'm not going to go into a lot of detail, just to say that for my initial analyses, I focused on the two largest morphotypes, the A and B morphotypes. I would get contents like this and pick out individual cells from these mixed gut contents, transfer the cells through wash solutions, and whenever I'd gathered a few thousand cells, I would lyse them and use PCR amplification to amplify the small subunit ribosomal RNA genes in that sample. When I started getting sequence information back from those amplicons, from those clones of amplicons, I found that the sequences I was obtaining looked bacterial in nature, and despite coming from different gut contents, they were very similar in sequence. From the A morphotype, I was getting two groups of phylogenetically related sequences, and from the B morphotype cells, I was getting a single sequence. This was a really exciting result, seeing these large organisms were nestled right in among the bacteria, but it was also a little bit unnerving because of the samples I was starting with. We know that it would be entirely possible that I was picking up some other bacterium that was living in the gastrointestinal tract and maybe just attached to the surface of these large cells, and maybe that's what I was picking up at the end of my processing. So I needed a way to confirm that the sequences I was obtaining were in fact coming from Epilepicium. And I did that using fluorescence in situ hybridization. I would take the sequences from these putative Epilepicium sequences and look for regions that were signatures for this group. Parts of the ribosomal RNA gene that were unique to these putative Epilepicium sequences and not found in other sequences in the database. I then designed oligonucleotide probes that would be complementary to the ribosomal RNA, labeled them with fluorescein, and then went back in gut contents and hybridized with those probes. And what I found was that those probes would specifically light up the ribosomes contained within Epilepicium cells, which confirmed that these sequences were in fact coming from Epilepicium and not some other bacterium. That allowed me to place Epilepicium in that larger phylogenetic framework, specifically in the bacteria in the low GC gram-positive bacteria phylum Firmicutes, and specifically related to members of the genus Clostridium. So if I zoom in on this region of the tree, This is the kind of phylogeny that I was generating from those sequences. Here are the two different clusters of sequences that I was getting from the A morphotype cells and the single cluster that I got from the B morphotype cells. Since this initial description, we've gone on to look at other of those large bacteria symbionts of surgeonfish and found that using fish, we could confirm that those different morphotypes that Kendall originally described form a coherent clade within the low GC gram-positive bacteria. Okay, I'm going to transition now and tell you a little bit about Metabacterium Polyspora, which is another model organism that my laboratory studies for maybe some obvious reasons. Metabacterium Polyspora was introduced to me by Carl Robineau, who had studied the DNA of Metabacterium Polyspora back in the 50s, and he said, this is a really interesting organism, looks very similar to your Epilepicium, maybe you should study it as well. And I totally agreed. So Metabacterium Polyspora was first described in 1913 by Edward Chiton and Charles Perard. It's a large bacterium, not quite as large as those giant epulos, but cells can reach lengths of 12 to 35 microns. This bacterium has the unusual ability to produce as many as 9 endospores per mother cell. This image is a phase contrast micrograph of the cecum contents of a guinea pig. And you can see all of these large cells with multiple phase bright endospores, as well as some of these large cells that are just simply phase bright. Those are all Metabacterium cells. Metabacterium bears a striking resemblance to some of the smaller epulos, such as the type F epulo that's found in the surgeonfish Acanthrus olivaceus. It's a fairly large cell, 15 to 60 microns long, and it was described as having the ability to produce as many as 7 internal daughter cells. So again, when I was a grad student, my task here was to see whether or not this morphological similarity between the F morphotypes, or epulos, and their daughter cell formation, and some of these Metabacterium were reflecting an evolutionary relationship between these groups of organisms. Again, we can't grow Metabacterium in pure culture, so I used some differential centrifugation methods to enrich for Metabacterium. I lysed them, used fish to confirm the source of the sequences that I obtained, and that allowed me to place Metabacterium in this phylogenetic context. And in fact, Metabacterium is one of the closest known relatives of the Epilepicium group. This was one of those aha moments and led to the hypothesis that internal offspring formation that was observed in Epilepicium may have arose from endospore formation. Other members of this group, of this particular clade, include Clostridium lenticellum and a Clostridium called strain PS7. These are pretty typical looking Clostridia. They're 1 to 4 microns in length, and they have the ability to produce a single dormant endospore. So my lab's been working towards trying to figure out how much of sporulation is conserved in Epilepicium and whether or not sporulation mechanisms are used to produce live internal offspring. What we use as the basis of our comparisons are the decades of information that have been gathered from research on the model organism Bacillus subtilis. Bacillus subtilis is a soil bacterium that has the ability to produce an endospore. It's a fantastic genetic system, and it's been extensively studied. I like to think of Bacillus subtilis as having 2 alternative lifestyles. When nutrients are plentiful and conditions are good, Bacillus subtilis can replicate its genetic material. It grows to about twice its starting size. It segregates that material into the 2 halves of the cell, and the cell divides. It continues this vegetative life cycle using binary fission for reproduction until conditions take a turn for the worse. When nutrients become limiting, Bacillus subtilis can cash in its chips. It can go through a developmental process to produce a highly resistant dormant cell, an endospore. In this case, Bacillus subtilis is starving for nutrients. One of the first things it does after it replicates its genetic material is it clamps down on the replication origins and prevents any additional chromosome replication from occurring. The cell then divides asymmetrically, producing a small cell called the forespore and a larger mother cell. As development progresses, that smaller cell is fully engulfed by the larger mother cell, eventually pinching it off as a free protoplast within the mother cell. At this stage, there's one chromosome copy within the forespore, and another chromosome copy is retained in the mother cell. Through the coordinated expression of genes in both of these chromosomes, that internalized cell is prepared for dormancy. Sporulation-specific proteins are produced, spore-specific peptidoglycan, the cortex is produced, and through the developmental process, the cell becomes very dehydrated, ATP content decreases, and eventually the mother cell lyses, releasing this mature dormant endospore. In this state, the cell can wait out those nutrient-deprived conditions for days or weeks, years, decades. Some people even say 1,000,000 years, although I'm not really confident in the data on those lengths of times. Although I do believe that that endospores can survive for decades and even hundreds of years. Even though this is a dormant cell, it still has the ability to sense changes in the environment. On the surface of the cell membrane are receptor proteins that are specifically designed to detect germinants, which are usually things like amino acids, nutrients that can support the growth of Bacillus subtilis. When those nutrients are sensed, this cell becomes reanimated. It rehydrates, it outgrows from the spore core, from the coat, and it can re-enter this vegetative life cycle. So you can imagine with Epilepicium, we think that this process probably occurs, but instead of becoming a dormant internal cell, if you stopped at this stage and allowed the internal cell to grow, you would essentially produce a daughter cell, an internal daughter cell. With Metabacterium, we saw an additional modification where the cell, instead of just dividing at one pole, it divides at both poles to allow for the production of multiple forespores. So I really don't have time to go into all of the data that supports this model, but this is what the Metabacterium life cycle looks like. It's very different from Bacillus subtilis, where binary fission is almost a vestige, and it can actually be bypassed altogether, where we've seen cells emerging from their spore coats that have immediately started to sporulate and appear to bypass any vegetative division cycle. As I said earlier, the cell divides at both poles, producing 2 forespores that then are competent to undergo division, producing multiple forespores that mature into multiple endospores. We think that this is an important means of reproduction that allows Metabacterium to produce a dormant, highly resistant cell type that allows it to cycle in and out of its host's gastrointestinal tract. It can then survive being, Metabacterium is a strict anaerobe, it can survive being outside in an oxygen-rich environment and also survive passage through the upper GI tract of its host. So Metabacterium constantly cycles in and out of its host's GI tract. For Epilepicium, we've studied development primarily in the B morphotype that I mentioned earlier and its host fish, Nasotonganus. One of the reasons we've chosen the B morphotype to study is that populations of this microbe in the gut are genetically much more homogeneous than any other epilep populations that we've looked at so far. It's also a really wonderful cell that's amenable to immunolocalization, and so we've used it for a lot of our cell biological studies. Again, I don't have time to go into the details of the data that goes into this life cycle, but based on a lot of data. We've been able to piece together the Epilipycium life cycle, which is shown here. And you can see that what's missing is binary fission. We've never seen any evidence that Epilipycium type B undergoes the traditional form of binary fission. And instead, it relies on this extreme polar division, engulfment of that polar cell, and growth of that internal offspring as its sole means of reproduction. We also have never seen any evidence of endospore formation in Epilipycium type B. We don't see phase-bright offspring being produced that are true endospores. And we think that drive to produce a dormant offspring has been relaxed in these populations because it appears that resident populations are established pretty early on in the life of a Nasotonganus, and those populations are maintained throughout the life of the fish. So they may not need to make that dormant cell. I certainly don't have time to go into this analysis, but I just wanted to tell you a little bit about some work we've been doing with the Epilopicium genome. We've been looking for evidence in the genome of type B to see if there are any sporulation genes that are conserved in the genome that may be functioning in internal offspring formation. So we went back to our touchstone, our huge amount of foundational information from Bacillus subtilis, where we know that there are 700 plus genes in the Bacillus subtilis genome that are differentially regulated as Bacillus subtilis enters sporulation and goes through this developmental process. We looked at the conservation of these genes in other known spore former genomes, eliminated all of the genes that are known to have some kind of a vegetative function, and specifically focused on genes that have a sporulation function. Of that 700 genes, this was work done by Dave Miller in my lab. Dave was able to whittle down that list to about 150 genes that are highly conserved in spore-forming bacteria that are specific for sporulation. And he found that only a small subset of those genes were conserved in Epilepicium. The genes that were conserved were involved in these early stages of asymmetric division and engulfment of the forespore. He found fewer and fewer genes that are required for making a highly dormant and highly resistant cell type. Fewer of those were conserved in Epilepicium. So far, we're gathering evidence that's supporting this model that live internal offspring that are produced in Epilepicium, that process evolved from endospore formation. This actually might be a good time for us to stop, and I'd like to take any questions you might have on that introductory material before I get into the next stage of the talk, where I'm going to focus more on large cell size and bacteria, large bacteria.
Thanks, Esther. So we can do questions through Twitter or through chat, or you can unmute your mic. Did we hear that? Questions through Twitter, chat, or unmute your mic are options for questions. Well, one of my general questions, I guess, because I was fascinated that guinea pigs might have, these large symbionts also, is do we have any idea about large symbionts in terms of gut structures and how abundant they are or frequent in terms of animals?
I think the gut environment is one of those new frontiers, and people are certainly looking at diverse guts, and with molecular methods, I'm not sure how often people are looking just using microscopy. So I think they're out there. There's an older paper that was published in the late 80s where a group of biologists saw Metabacterium-like bacteria in some of their rodent colonies, and they said, oh, I wonder how prevalent these are. And so they started looking at some exotic rodents, and they found Metabacteria-like cells in agouti-pacas and capybaras and quite a variety of rodents, and they all had this similar characteristic of being these fairly conspicuous cells that are fairly large that produce multiple endospores. So, you know, for a microbiologist who uses a phase contrast microscope all the time, I would think, wow, these must be like I don't know, fireworks, you know? How could you miss them? But I think we're still discovering microbial diversity. You know, we're still discovering really surprising and diverse organisms that are practically under our nose. So there is a bit of literature from the older, you know, like the 50s, that describes large bacteria in a variety of different GI tracts, but they really haven't been studied beyond just their morphology.
So we have two more questions. I have a question, but I'll first ask a question from the internet that came in from Denmark, presuming Ian's at home. So Ian Marshall from Bo Jurgensen's lab would like to know, how about large bacteria in the human gut? What's the largest bacterium in there?
Oh, wow, that's a great question, and I don't know that I can answer that. I've never seen one of those. When you're a graduate student, I'll back up a little bit. When you're a graduate student, you kind of have time to chase really crazy ideas. And so one of the really crazy ideas was, can we find epulos in other systems like maybe humans? Getting human gut samples at the time was kind of challenging for us. I don't know that anybody has ever seen large bacteria in the guts of humans, although we do see a lot of members of the Lachnospiraceae, which is where Epilepicium falls. So I don't think there are any exceptionally large bacteria in the guts of humans. So they're probably all ranging about, you know, the normal bacterial size, from less than a micron in length or in diameter to just a few microns, maybe 5 microns in length.
Okay, so Caleb has asked, is there any idea how the tear in the envelope of the mother cell is repaired? Is it a directed process or more general stress response?
That's also a great question. And I've gone through this introductory material so fast that really important details sometimes I skip over. This is a terminal process. When the mother cell lyses and the offspring are released, the mother cell is dead. What you're left with is this husk of the envelope of the mother cell. We actually think that the mother cell goes through a programmed cell death at the terminal, kind of at the end stages of daughter cell development. The most obvious change that we see is DNA in the mother cell diminishes. So we think that the DNA is being broken down and maybe shuttled into the developing offspring as either oligos or nucleotides that can be used for growth and development of the offspring. So the mother cell is never repaired, it's killed in the process.
Okay, and then we've got another question that came in. Has anyone sequenced the genome? What insights has it given us as far as their metabolism? Was in terms of a host? Oh, I'm sorry. Well, I guess the question is, are there any metabolisms that have a connectivity with the host in terms of the processing of the fish, like fish gut metabolism?
Yeah, that's we've been working on the Epilepicium type B genome. It's still fragmentary, but we have a really reasonably good draft. It's in about 60 contigs right now. One of the big features in the Epilepicium genome is that it codes for a lot of polysaccharide degrading enzymes, and those polysaccharides are polysaccharides that you would see in algae that the fish is consuming. So things like laminarinases are coded for in the genome, and other polysaccharide degrading enzymes. So we think that epulos are breaking down these polysaccharides that otherwise would just pass through the gut of the surgeon fish if it weren't for microbial degradation of those polysaccharides. The simple sugars are then fermented into volatile fatty acids, and there have been studies that showed that those volatile fatty acids, things like acetate, are taken up by the fish and used for energy, as a source of energy. We're currently working on a a project to look at nitrogen metabolism, and we think that epilose might be an interesting model for allowing a symbiotic population to live in the gut of a host, but periodically lyse part of its cell whenever the mother cell lyses and release some nutrients into the gut lumen that may be taken up and used by the fish. So we think that the daily lysis of the Epilepicium mother cell might be a mechanism that releases proteins or possibly some nucleic acids, some organic nitrogen sources that the fish or other microbes in the gut can use.
Okay, and then I have to just ask before you finish, but I might distract you with this question. But we are hearing about gut studies ad nauseum right now.
Yeah.
But they're all fecal analysis of gut flora for the majority of gut studies. And so what you're talking about is an actual gut, right? And you're saying that the fish doesn't lose its population throughout the fish's life cycle.
Yes.
So I was wondering, as a person who studies gut symbionts, how do you feel about this idea that you can do fecal analysis and backtrack to a gut community? And what are we missing if we're not doing microscopy on human guts?
I think it's hard to say what we're missing, but I know from fish guts, which are, highly accessible because we can open up the fish, we can sacrifice the fish and start looking at different parts of the gut and looking at the populations of microbes in that gut. Like humans, we have lower densities of bacteria in the anterior gut, but those numbers increase as we go more posteriorly in a surgeonfish. We see populations coming and going. So there are regions of the gut where you find dense populations of epilose. They're the most conspicuous, so they're kind of the easy ones to spot. If you took a gut and laid it out from a surgeonfish that had epilose, and you laid it out in a line on your desk about 2/3 away from the stomach, about 2/3 of the length away from the stomach going posteriorly is where you'd find really dense populations of epilose, and then those numbers trail off as you get towards the anus. So that's just one group of microorganisms. When we look at sequences, when we do community surveys, we see populations, and when we sample along the length of the gut, we see populations coming and going. So I think of the gut as a very dynamic continuum that is different in different regions. Certainly the microorganisms in those communities are seeing different environments and are doing different things. How that impacts the host, we may not be totally seeing the full picture in the anterior intestine of humans because we're looking at fecal bacteria. Certainly that's where the colon is the densest populations, and they're metabolically important, but we may not be seeing more subtle, smaller populations that predominate in the upper GI tract, the more interior portions. It's just harder to sample,
Oh, completely understand. I'm saying, in the interest of time, why don't we hold questions for now, and then we'll keep talking for the seminar, and we'll do questions after the end of the seminar.
Sounds good. Okay, so let me pick up here with, this is one of my favorite photos, at least favorite for now. You all recognize this as an Eppendorf tube, and all of the little flakes that you see in this Eppendorf tube are epilose. So this is just a tube of epilose that have been picked out of the gut of Nasotonganus. Epilepicium, and even more so, this beautiful bacterium, Thio Margarita nimimbiensis, are enormous microorganisms. Thio Margarita was first described in 1999. Again, it's an organism that's out there, it's abundant in certain environments, and it's highly conspicuous. The micrograph on the right, I should say that I pulled both of these micrographs off the web, and they're both from Heidi Schultz-Volt's lab. She was one of the original discoverers and people who described Thio Margarita. The micrograph on the right is probably just taken from a dissecting microscope, looking at some sediment samples, and these beautiful chains of cells that look like a string of pearls. And then these enormous individuals, you can see the scale bar here is 1 millimeter. This guy's at least 500 microns long. You can see these cells with the naked eye, and under the microscope they're quite beautiful. These spherical cells contain tiny spheres inside their cytoplasm that are sulfur granules, and those sulfur granules reflect light, which is why these look like a string of pearls, and they give Thio Margarita their name, the sulfur pearl of Namibia. So we've got these enormous bacteria. We've actually known about large bacteria for more than 100 years. There are reports of large, either free-living microbes like Spirocheta plicatus, which I always slaughter its name, but large free-living spirochetes and also large gut microbes described in the literature more than 100 years ago. But all of these large bacteria all sort of had a very similar cell structure that fit in our concept of what a bacterium should be. So all of these large bacteria, even though they could be several hundred microns in length, were no more than 2 microns in diameter. This allowed no part of the cytoplasm to be very far away from the external environment. So let's back up for a second and think about bacteria and our concept of bacteria. We think of bacteria as being very simple cells, and if you look back in older textbooks and older literature, you might even see them described as bags of enzymes and nucleic acids. So they were just considered disorganized, very simple cells. And because of their lack of intracellular organization, which became really obvious in the 50s when we started looking at thin sections using electron microscopy, we saw all this beautiful intracellular membrane structures and organelles in eukaryotic cells, but we didn't see this in bacteria. So we thought we knew everything we needed to know about the bacterium, that it was simple and disorganized. And because of that, it relies on diffusion to get nutrients from its environment and also to move biomolecules throughout the cytoplasm. Diffusion is a very efficient process to move a molecule from point A to point B, as long as point A and B are very close together, only a few microns at the most. You get almost instantaneous movement. But as you start increasing that distance, diffusion becomes more and more unreliable as a means of transporting molecules from point A to point B. So bacteria had to stay small so that they had a large surface area relative to their cell volume so that they could encounter nutrients and take them up and use them in an efficient process in a timely manner. So big bacteria all seem to fit in this by having very thin cytoplasm. Even Thio Margarita, which is an amazing cell, 98% of its volume is taken up by a central fluid-filled vacuole. And the cytoplasm, the active cytoplasm where the DNA is, where enzymes are, is found in a thin layer just under the cytoplasmic membrane that's no more than 2 microns deep. Epilepicium seemed to be exceptional in that you could have active cytoplasm that could be as much as 20 microns away from the external environment. So we were really keenly interested in studying features of Epilepicium that may have been modified or maybe even invented to allow it to support this large cytoplasmic volume relative to the surface area. You can get some hints by looking at electron micrographs of large epulos. This is a micrograph that we published some time ago showing a cross-section through a tiny portion of the mother cell. To orient you, this is the envelope of the mother cell, this is exterior of the cell, and interior. One of the most striking features whenever you look at EMs of epulos is this highly enfolded internal membrane system. We think that that's important, and it essentially increases the membrane surface area of the cell by just having lots of invaginations that we think are probably sites for transport proteins to facilitate movement of molecules in and out of the cell. Just beneath the cytoplasmic membrane is this layer of nucleoids, which is going to be the feature that we'll talk about the most in just a few minutes. Outside of the cell, you can see this layer of broken filaments. This large structure here is a more typical bacterium that's stuck in the layer of flagella that are on the surface of Epilepicium. We think flagella might also play a role in allowing this cell to be so big. I'm going to show you another one of these movies down an eyepiece, taken down an eyepiece, of some of the J morphotype epulos that you can see lying here under a microscope cover slip, between a cover slip and a slide. So they're kind of stuck, but their flagella are still beating, and you can see that they're causing a lot of turbulence in the surrounding environment. We think that this might be important for Epilepicium, and they use their flagella similar to what some ciliates do, and they use it to refresh the immediate environment, which could help overcome relying solely on diffusion to move waste products away from the cell and to bring nutrients closer to the cell. So we're currently interested in studying this, but I don't have any really great data to show you at this time. But one thing that we do have a really good handle on is the DNA in Epilepicium. This is a fluorescent micrograph of Epilepicium stained with the DNA dye DAPI. So everything that you see in this micrograph that's blue is DNA. And I hope you can, now that you've seen epulos many times, you can tell that this is an Epilepicium mother cell with 2 large offspring. Here's a single offspring, and here's one with 3. Also in this micrograph, since this was just total gut contents, you can see this fellow up here, which is a large ciliate that also inhabits the nasotongonus intestinal tract. This guy is called Ballontidium jocularum. And all of these little dots, prominent dots that you see here and down here, those are the nuclei of some flagellates that live in the GI tract. So comparing the amount of DNA in these large bacteria to the nuclei of ciliates and flagellates, you can see that there's a ton of DNA in an individual epulo. We estimated from doing some really simple experiments of just picking a number of cells, lysing them, and then extracting the total DNA and quantifying it. We know that large epulos like this with 2 large offspring cells contain on average about 250 picograms of DNA. For comparison, our human cells that are diploid contain about 6 picograms of DNA, and E. coli K12, its genome is about 4.6 femtograms of DNA. So the first question we had was, what is this DNA made of? Is it representing an enormous genome, or is it something else? Is it maybe a bacterial-sized genome that's there in lots and lots of copies? We were pretty sure that it wasn't just a tremendously large genome, because we knew that during that initial polar division event, only a tiny portion of the DNA from a mother cell is partitioned into that newly formed offspring cell. So we figured that this tiny amount of DNA was probably at least one, if not multiple, genome equivalents. We found that we could use quantitative PCR to, well, actually we could just use PCR. If we took an individual Epilepicium cell, put it into a PCR tube, we found that we could amplify genes using PCR pretty readily. So we used this to our advantage and used quantitative PCR to enumerate gene copy numbers of specific gene targets in Epilepicium cells. We targeted the extremes of size and development, looking at 2 different populations of cells, large cells with large offspring. We also tried to enumerate gene copy number in small cells with small offspring. These are just a couple, again, DAPI stained cells representing those groups of those populations. The first gene that we focused in on was FTSC. FTSC is a gene that's important for producing a protein that serves as a scaffold for the cell division machinery to assemble on at the site of division. FTSC is normally found in a single copy per chromosome in most bacteria. There are a couple examples where there are two paralogs of FTSZ in a genome, but in low GC gram-positive bacteria, there's usually just one copy per genome. So when we targeted that, we found, actually what we would do is we would take individual cells, take them out of the total gut contents, photograph them, and then take that individual, put it in a qPCR tube, and enumerate FTSZ gene copy number. From the photograph, we could measure the length and the width of the cell, and from those measurements, we could estimate cell volume. We then plotted cell volume versus gene copy number for FTSZ. The pink dots represent small cells with small offspring, and then these blue diamonds represent large cells. So each individual point is a single cell where the volume and gene copy number have been plotted. And we see this pretty good linear relationship between cell volume and gene copy number. However, it also represents how much diversity we would see in a single population of epulos, size diversity and also genome or gene copy diversity. What our ultimate goal was to use quantitative PCR to try to figure out if individual single copy genes showed different proportions in an individual, which may indicate that regions of the genome were preferentially amplified. So we expanded our survey, again using individual cells. We looked at not only FTSZ, but also DNA-A, REC-A, and we had the 16S ribosomal RNA gene clone, so we looked at it as well. Unfortunately, we saw a heck of a lot of diversity in gene copy numbers for all of these markers. So it was hard to say whether or not portions of the genome were preferentially amplified. Instead, we took a different tack, and we took populations of cells, isolated them, extracted their DNA, and then enumerated these different single genome copy markers in a fixed amount of genomic DNA. When we did this, we found that the gene copy numbers for the same amount of DNA extracted from Epilepicium cell populations in large cells with large offspring, we essentially got very similar, statistically identical numbers of each of these three single copy genes. When we looked at the DNA from small cells, we found that DNA-A had a statistically significantly larger number of genome copies or gene copies than the other two markers, but that kind of made sense to us. DNA-A is, in most bacteria, it's linked to the origin of replication. So you can imagine in these small cells that are actively replicating their chromosome that you may have chromosomes that are partially replicated, and DNA-A might be more abundant because of those multiple origins of replication. What we concluded from this data is that it all supported a model that Epilepicium is extremely polyploid, and that it contains probably an approximately 4 megabase genome that is there in just tens of thousands, even hundreds of thousands of copies. This is another pair of micrographs that I really like because it gives you a three-dimensional sense of where the DNA is in Epilepicium. These are a single DAPI stained cell, but a micrograph with a focal plane going through a medial section of the cell, and then this section is just under the cytoplasmic membrane. So you can see the DNA forms this network just under the cytoplasmic membrane that totally encases the cell. These polar structures are the start of the next round of offspring. Betsy Hutchison, who is a postdoc in my lab, has been using DNA fish to label origins of replication, or probes that would light up origins of replication, and also looking at comparing the localization of two different chromosomal regions in single Epilepicium cells. I'm not going to talk a lot about this today, but I just wanted to show you this really beautiful micrograph where Betsy has labeled two different regions of the chromosome. In green, a green probe as well as a red probe. And I hope you can get a sense from this. This is actually a compile micrograph of multiple focal planes through the thickness of an Epilepicium cell. And I hope you can get a sense whenever you look solely at the green markers or solely at the red markers that these chromosomes are fairly regularly placed around the periphery of the cell. And we think that this is an important adaptation that allows Epilepicium to support such a large volume of cytoplasm. These genome copies, these chromosomes, are located just under the cytoplasmic membrane, but throughout the periphery of the cell, which allows epulos to respond to environmental stimuli immediately. Instead of having a centralized nucleoid that you might see in most typical bacteria, if epulos had that, it would take such a long time for the signals detected on the surface of the cell for signaling to reach the nucleoid, and then for genes being expressed, and then those products diffusing back out to the periphery of the cytoplasm where they might be functioning. So we think being extremely polyploid is one of Epilepicium's tricks. It's just taken the simple building blocks of this quote-unquote simple cell and just amplified it to allow it to support a large cell size. This is, again, a DAPI stain of Epilepicium, and beside it is a photograph that, that Verena Salmon has taken of a Thio Margarita cell. I've got to throw in a disclaimer here. This is a pretty small Thio Margarita cell, but the reason we chose this small cell was that it gave a much better global picture of the DNA in a particular cell. And you can see that there are multiple nucleoids around, again, the periphery of the cell, and we think that Thio Margarita has a very similar mechanism as Epilepicium, having lots and lots of nucleoids, being highly polyploid, and that's one of the ways that it can also maintain its tremendous amount of cytoplasm. Just looking at this cell, you can see these are shown at the same magnification, almost the same magnification. I think that each of these nucleoids is probably multiple copies of the chromosome. Verena is going to be coming to my lab later this year, and we're going to start investigating chromosome copy number in Thio Margarita cells. So stay tuned. I mentioned earlier that we think that the mother cell undergoes a programmed cell death as part of the developmental process. And I was highly interested in whether or not chromosomes in the mother cell were dismantled and then those nucleotides shuttled into the offspring cell as a source of building blocks to allow the cell to develop, to support the development of the internal offspring cell. So we looked into a variety of different methods to try to pulse label genetic material to see if we could chase it into different parts of the cell, from the mother cell specifically into the offspring. At first we were thinking about using radioactivity, but that seemed like a really daunting task because radioactivity, using it out in a field station where they're not really set up to use radioactive substances, seemed like a really huge barrier. So instead we turned to a reagent that's commonly used by eukaryotic cell biologists to label newly replicated DNA. And this reagent is called BRDU, bromodeoxyuridine. It's a nucleotide analog that is taken up by lots of different cell types, both eukaryotic cells and bacterial cells. It's taken up and incorporated, phosphorylated and incorporated into replicating DNA very readily. But because it's got this bromide attached to it, it's got a great handle that you can use antibodies that will specifically identify bromodeoxyuridine incorporated into DNA. So we used this reagent. This was work done by Becca Ward in my lab. Becca used BRDU to try to label Epilepicium cells in the field. One of her most successful experiments was looking at a population of cells that were about this stage of development, where offspring cells had been formed and they have started to grow within the mother cell. So at this stage of development, no additional offspring cells would be initiated. So Becca just simply pulse labeled these cells in the GI tract of a surgeonfish with BRDU, and then looked to see where replication was occurring. As we expected, we saw replication occurring in the DNA of offspring cells, but what was surprising to us was that replication was still occurring in the chromosome copies in the mother cell. And you can see that in this fluorescence micrograph showing the immunolocalization of BRDU in an Epilepicium cell. This was fascinating because we know that these chromosome copies are never going to be inherited by an offspring cell. So they're replicating, but they're not going to be inherited. We interpreted this result as being essential. It's essential for these cells to replicate these quote-unquote somatic genomes so that we have enough genomic material to support the metabolic needs of this mother cell that in turn is supporting the growth and development of offspring cells. Becca did a similar analysis with Metabacterium Polyspora, where she labeled Metabacterium cells in the gastrointestinal tract of guinea pigs. And in this, she almost saw the reverse localization pattern, where developing 4 spores were taking up BRDU and incorporating the analog into newly replicated chromosomes. So for this study, we interpreted this as representing active DNA replication throughout the sporulation process, which is in stark contrast to what we see in Bacillus subtilis, where it shuts down replication during sporulation. We think that this is important so that the cells can be loaded up with multiple chromosome copies that then can be partitioned into the developing 4 spores. And in Epilepicium, we saw this replication primarily in the mother cell. This may have just been due to first come, first served, where the BRDU was being transported into the mother cell and preferentially used, because we really saw intense staining in the mother cell compared to the offspring. But we saw this really interesting aspect of somatic chromosomes being replicated, even though that they would never be inherited by an offspring cell. Okay, so extreme polyploidy. We think that it's important for epulos to be extremely polyploid because it allows them to maintain their metabolic activity to support this large, highly motile cell. It also allows them, this large cell size, allows them to maintain their position in the gut. We've clocked epulos, large epulos, as swimming as fast as 400 microns per second, and we know that they migrate on a daily basis anteriorly at night so that they aren't eliminated from the gut, let's say it that way. We also noticed from some studies that we did looking at single cells and the genes in single cells that Epilepicium appears to be able to tolerate genomic defects that otherwise might prove to be lethal in a smaller bacterium. Specifically, we know that Epilepicium contains poly-A tracks within coding regions that are longer than what's usually tolerated by DNA and RNA polymerases. In essential genes like DNA-A, we see poly-A tracks that are 10 nucleotides in length, which is a length that can't be really accurately read by polymerases very well. This is a very rare feature that seems to be found either in organisms that have found a mechanism to repair these defects or in highly polyploid organisms. We also think that being highly polyploid allows Epilepicium to gain advantages that are normally only seen in eukaryotic cells and social microbes. I like to think of epulos as being almost like a colony of cells, tens of thousands of cells that just never separated, never divided from one another. Certainly being large is nice in an intestinal environment and probably allows you to avoid predation, but it doesn't totally allow you to avoid predation this fellow over on the left-hand side is a Bacillus cell that has recently eaten an Epilipycium cell. So you're not totally saved. Okay, just to summarize quickly, because I'm way over time, Epilepicium species, the smaller Epulos, and Metabacterium Polyspora are all intestinal symbionts. We think that it's a specific, mutually beneficial symbiotic association where the fish benefits because these microorganisms are aiding in the digestion of polysaccharides primarily. The symbiont probably benefits by living in a very predictable environment with regular access to nutrients, regular predictable access to nutrients. The production of intracellular offspring appears to be derived from endospore formation, and we're currently looking at the genomes of Epilopicium species to see how this process has been modified for different forms of reproductive strategies. Large cell size is supported by extreme polyploidy. Genome copies in Epilepicium have now taken on both somatic and germline roles, where about 1% of the chromosomes is passed on to offsprings, and that other 99% appears to perform the same role as our body cells, just to support this reproductive function and development of offspring. And finally, polyploidy is found in diverse bacteria, and so I'd like to throw this idea out at you that polyploidy is not quite so rare as people think it is. It's found, there are great examples of polyploid bacteria in many of the major phyla of bacteria, and I think that ploidy is a target for selection. In environments where nutrients may be more predictable, such as a gut environment, polyploidy may be the norm because nutrients are not limiting, and polyploidy has advantages over just being having a single copy of the chromosome. So then my final slide is just acknowledgments, thanking longtime collaborators Kendall Clements, Howard Choate, Lynn Montgomery. I want to thank Verena for giving me some really great micrographs, and I'm looking forward to working with her in the future. And I'm just listing a number of the people in my lab, graduate students, postdocs who've done a lot of the work with Epilepicium and provided some really amazing insights into the biology of this organism. Listed just a few current undergraduates in the lab. I want to thank the fish, of course, and my funding sources, which are the NSF and Cornell University. So with that, I'd like to take any questions you might have. And thank you for hanging in there with my technical difficulties, and yeah, I'll take any questions.
All right. Well, as we wait for folks online to type in a... Oh, am I actually talking? I never know how fast my microphone turns off. Okay. So, as we wait for folks to ask questions online, if anyone in the chat has a question, please turn on your microphone and ask it. Or you can type and I will try to read it. There was a leftover question I guess I can ask for you. Has anyone looked at the guts of these fish compared to the fecal matter? And you mentioned that the fish actually move during the day, so I'm guessing there might be more there than you had mentioned.
Yeah, so very few. There have been very few studies, and there have been some misinterpretations of what's in the fecal matter compared to what's in the gut. So some of the epulas that we're studying actively in the lab that I didn't talk about today are the smaller J morphotypes, those really long filamentous organisms, and also the C morphotype, which looks a lot like the B morphotype, but it produces a true endospore. The interesting thing about these populations that are found in both Nasounicornis and Nasoliteratus is that they form endospores, and they do this on a regular basis, where endospores are seen in the GI tract but only seen at night. When we look at fish, these fish, if we hold them in captivity, we find that overnight, if they do defecate, you know, it's a little bit of an unusual situation because they're probably stressed out. They're sitting in a in an aquarium instead of being out in their little hidey holes in the reef and sleeping. But if we collect their fecal material, we do see epilospores in their fecal material that they've shed overnight. So it kind of depends on the organism. We definitely see spore-forming epulos regularly in the feces of some surgeonfish. Nasotonganus, even though it's a great model system, it's a challenging model system because it's such a large open water fish. It's hard to capture and maintain in aquaria. Surgeonfish get their name because they've got spines on their tails that are thought to be like surgeons' lancets, and so they're kind of a difficult species to handle. And so you may see them in aquaria, but a lot of times they're captured as smaller fish and brought to the aquaria as small fish, and they may not have all of their epulos in their guts. So that was my very long-winded question or answer to the content of feces varies depending on what surgeonfish you're looking at and what time of day you collect those feces, and it may or may not well represent what's in the GI tract.
All right. And another question was, you mentioned the motility, and so is there an estimate for how many flagella these organisms would have?
Oh, that's a great question, and I don't have a good answer for it because it's really challenging to count them. Flagella are kind of brittle. They easily break off, and so I really haven't tried to fix cells and bring them back to the lab to do any quantitative measures by SEM or by TEM. We really need to do some serial sectioning, I think, to get a better sense of the cell structure and also the flagellar arrangement. I've not tried to estimate it, although I think I probably could from some of the published micrographs. I'm going to guess there are thousands easily, maybe tens of thousands.
All right. Any other one in the Hangout have any other questions or coming through the internet? This has been fabulous, and I love the fact that you gave history and overview because I think a lot of us will use this as a teaching tool in the future. So I think Lizzie has a question.
Yeah, I do. This is Lizzie Wilbanks from UC Davis. Thanks, Esther. This was awesome. It was really cool.
Thanks.
I had a question. I was curious that you said they migrate every night. What do you think they're sensing to know when it's night? Do you have a sense from looking at the genome of any sort of sensors or chemotaxis or something like that that they might be using to cue in to different times when it's time to move? Yeah,
absolutely. Epulos have a number of what look like methyl-accepting chemotaxis proteins, or at least type B does. I had an undergraduate working on a project to try to bring those MCPs into a Bacillus subtilis to see if we could figure out what the various MCPs might be sensing, but she graduated before we got that system up and running. That's a great question, and we're currently heading into a project to start looking at what oscillates, what maybe the metabolome is. Maybe that's the sexy word to use, but we're interested in things like, do volatile fatty acid concentrations ebb and flow during the day, and that represents, active fermentation maybe happening late in the day when the gut is full and then diminishing as the nutrients are fermented. We're also interested in other chemicals that might be coming and going, and whether or not epulos are sensing those signals to maintain their position in the gut or to alter their metabolism. So that's an ongoing question, and I don't have a good answer for it, but we definitely know from the genome that there's that potential for epulos to use MCPs to chemotax.
Awesome. Well, we'll stay tuned.
Thanks.
All right, any other questions? All right, I don't see any, but I would like to thank Esther on behalf of everyone watching. We had somewhere around 20 people in a real-time environment, and we will likely have more watching the seminar afterwards in the recording.
Nice.
And so, yes, it's been fabulous having everyone here. We'll do another talk in 2 weeks, and because it is sorry, I'm clicking through things here. We have to do the appropriate seminar ending with the applause. Oh, my double.
Thank you. Thank you very much.
So thank you, Esther, and we will see you guys in 2 weeks.
All right. Thanks, everybody.